My name is Mallory Hansen, and today I will be presenting on the process of intramembranous ossification. Intramembranous ossification is defined as the development of bone from tissue or membrane, as in the formation of the skull, and typically starts in the 9th week of pregnancy. During fetal development, intramembranous ossification results in the formation of 3 different types of bones: flat bones in the skull, the mandible, and the clavicle. Intramembranous ossification takes place in fibrous connective tissue. In the connective tissue, the mesenchymal stem cells cluster so that they can begin replicating. This depicts the replicated cells. After they replicate, they differentiate, becoming a bone-forming cell called an osteoblast, which releases uncalcified bone matrix called osteoid, which is represented by the red. The osteoid is then calcified with calcium salts and other minerals like alkaline phosphatase, which is an enzyme that helps speed up this reaction. Osteoblasts, when surrounded by calcified bone matrix, can differentiate even more into osteocytes, which are housed in the lacunae, which is represented by the black. Ossification takes place in an ossification center, represented by the big blue circle. Needle-like structures called spicules begin to form. They grow until they meet in the middle and fuse together. Because mature bone is highly vascularized, the spicules will grow around the blood cells, which allow nutrient exchange and waste exchange during these crucial developmental stages. The bone initially formed by these growing spicules is known as spongy bone because of the latticework structure it forms. Then, through the process of bone remodeling or the breakdown of mature bone in order to rebuild new bone, the osteocytes at the edge of the spongy bone reorganize into osteons, which are the basic building blocks for compact bone, hence creating compact bone. In another form of bone remodeling, the middle section of spongy bone can be removed, which creates the medullary cavity or the central cavity of the bone shaft. The final structure to complete the bone is the fibrous connective tissue that is wrapped around the bone, known as the periosteum. In the tissue, more osteoblasts can be produced, which allows for growth in the width, known as appositional growth. Let's review. We began with mesenchymal stem cells, which replicate and then differentiate into osteoblasts, which build bone, and release uncalcified bone matrix. After the matrix is calcified, the cells differentiate again into osteocytes, which break down. The starting bone that is created is spongy bone, which can then be remodeled into compact bone. Bones created through intramembranous ossification include flat bones of the skull, the mandible, and the clavicle. Thank you for watching. My name is Mallory Hansen, and this is my presentation on intramembranous ossification.